National 5 biology students, let's do this past paper question together. So this one says pepsin is an enzyme involved in the digestion of proteins, which is a degradation reaction. The diagram represents 3 stages that occur in this reaction. Using the letters from the diagram, put the stages into the correct order to show this degradation reaction. So a degradation reaction is where we break down a large molecule into smaller molecules, okay? So we start with a large molecule and we end with smaller molecules. So that should help us put these into order, okay? Hopefully we understand then that Q must be the first stage because we've got the substrate, the larger molecule, going into the active site there. Then R has to be the middle stage because we've got the enzyme substrate complex and the reaction taking place where the large molecule has been broken down into smaller ones. And then P will be our final stage. And the reason for that is because our smaller molecules have been made via this degradation reaction and are now being released. The products are released from the active site. And that's what shows that the reaction is now over. Okay, so Q, R and P. Describe the feature of pepsin that allows it to bind to only one substrate. So the reason it can only bind to one substrate is because the active site, and this is well worth memorising this sentence, is complementary. Or you could use the word specific instead of complementary. Either one is acceptable. It's specific to one substrate, or to this substrate. Okay, I should probably say to this substrate because that's what the question's asking about. Okay, let's go on to part B. The graph shows the results of an experiment to investigate the effect of pH on pepsin activity. Okay, so it says, use the graph to identify the optimum pH of pepsin. Now, let's have a look at what the results are showing before we jump to any conclusions here. This is the protein remaining after 24 hours. So this is how much protein is left, okay? Now we're digesting that protein. So the more protein that's left, the less reaction has occurred. So that means it's least active if it's got a high number of protein left. Whereas if there's less protein left, that suggests that the enzyme has been more active, and therefore that suggests the optimum, because we know the optimum means where the enzyme is most active, okay? And therefore, where it's most active is where least protein remains, and that's going to be at this point here, okay? And if we go down to our scale here, that point there is going to be 2.2, 2.4, okay? Hope that makes sense how we got there, folks. And part C, enzymes can be denatured by changes in pH. Give a reason why the rate of reaction will be affected. Okay, the reason why is because the active site, when it is denatured, changes shape. Okay, and therefore the substrate can no longer bind. Which means no reaction will take place, okay? So the reaction will, or very few reactions will take place, which is why the rate of reaction will go down to pretty much 0. Okay, and we can see for pH 7 and pH 8 here in this particular example, the protein remaining is 100%, i.e. there's no reaction has taken place at really high pHs, and the reaction obviously is slower at 6 and 5 than it was at 2.4. Okay, so that's what we're looking for that one. Hope that helps, folks. See you in the next one.